Good morning, everyone. I'm Robert Chipschooley from the University of California, San Diego, here with my colleague Graham Hatfill from the University of Pittsburgh to talk to you today we hope, a bidirectional symposium about bacteriophages, which are increasingly being used to treat multi-drug resistant and difficult to treat bacterial infections. The goal today is in Graham's section of this to give you a sense of the biology of bacteriophages, what they are, where they come from, what they do. And I'll talk a bit about some of the current clinical applications and challenges to moving them from being used primarily in anecdotal one-off case situations to full access as an antimicrobial that would be added to the shelf along with traditional antibiotics. We hope that during the course of the seminar, you'll come up with some questions and discussions that you want to have after each of our conversations. And we also hope that you will think about ways that we can move this field forward. It's clearly something that we think is going to be very important in our efforts to better control multi-drug resistant bacterial infections. So with no further ado, let's move forward. So these are the members of the International AIDS Society USA Board of Directors. This symposium is sponsored by the IAS-USA. Next slide. And Dr. Hatfill and I, our financial relationships are listed here and will be listed during our presentations as well. Next. Those of you who are looking for CME credits will be able to receive 1.25 CME credits of various categories, depending on which ones matter most to you. There'll be instructions about how to do this later. And as I've already said, the activity is supported by the International Antiviral Society USA. Next. So to navigate this activity, there'll be a separate window that'll show you some poll questions we'll ask from time to time. You can choose your response for the poll, and we'll display the results after the poll closes. You can submit questions through the Q&A button. We may not be able to answer all the questions. We'll try to get to as many as we can, and we'll try to get offline to some of those that we don't get to during the hour and a quarter that we have with you. And please, the chat is open. Feel free to use this to discuss things you're hearing with other attendees. Don't suggest and submit questions through chat because we won't be able to see them. We'll only be looking at the Q&A. Next. So let me now introduce you to Graham Hatfill, who is the Eberly Professor of Biology at the University of Pittsburgh. Graham is a pioneer in bacteriophage research and has been working for almost his entire career on the basic biology of bacteriophages, and particularly those that prey on mycobacteria. So let me turn this over and let Graham give you an overview about how phages interact with their prey and vice versa. Graham?
Thank you very much, Chip. It's a delight to be here, and I'm looking forward to sharing with you some of the basic principles and background information about bacteriophages, and this will hopefully nicely set us up for then hearing from Chip about some of the more clinical aspects. So first of all, this is just a declaration of the financial relationships. The principal learning objectives are shown here, but I would also just really summarize this as the key objective is to understand some of the basic background information about bacteriophages, especially in those aspects that are relevant to their clinical utility and the prospects for clinical utility. I have one question that we'll focus on here, and so this is the pretest question, and I will let you read that question and submit your answers. This will then be presented as a post-test question at the end of this section. just give it a few more moments to submit your answers. Thank you, and we will proceed. Okay. So antimicrobial resistance has become a major concern. It's been a concern for some time, really, ever since antibiotics were introduced to treat bacterial infections, for which they are, generally speaking, very effective. It was clear that bacteria could become resistant quite readily to antibiotics, and that concern has become ever of more greater concern, and there are these estimates that this is going to be a very severe problem in another few years. And because of this, there's been a quite an urgent need to consider new types of therapies, the types of therapies that will effectively kill bacteria in a human infection. And bacteriophages have long been recognized as having that potential, although therapeutically the potential has yet to be fully realized, a topic that we are all, of course, quite interested in. So what is a bacteriophage? A bacteriophage is a virus. It has the properties of other viruses. It does not replicate by itself. It requires a host in which to replicate. And a bacteriophage, or phage for short, is a virus that infects bacteria. They're small, common with other viruses, so you don't see them under the light microscope, but you can see them under electron microscope, and they come in many different shapes and sizes. But the micrographs that I'm showing you here show some of the more common types of morphologies, the ways in which they look in the electron microscope. They're relatively simple. Many of them have a head, sometimes called a capsid, which contains the genetic material, commonly DNA, and sometimes, as in these examples, there is a tail attached, and the tail is what is able to recognize the bacterium host. There is great specificity of bacteriophages for their bacteria. So there is no one bacteriophage that kills and infects all types of bacteria. Bacteriophages tend to be specific for a bacterial genus, more commonly a species of bacteria, and even more commonly than that, the specificity is often constrained to just a small number of clinical isolates within a species of bacterium. And therefore, this specificity is a chief concern or facet in thinking about therapeutic utility, because any phage that you want to use therapeutically needs to be able to infect and kill the particular clinical isolates of bacteria that make people sick. Phages, generally speaking, can be grouped into those that are referred to as lytic. As we'll see, they infect bacteria, they replicate and kill the bacteria. But there is another major class of phages which are referred to as being temperate. And those phages, as such, are more problematic for use in clinical cases because the infection by the phage doesn't always lead to the death of the bacterium. And we'll discuss how that can be addressed experimentally. The phages out there in the environment are estimated to be vast in terms of their population, 10 to the power of 31 total phage particles, which makes them, by the numbers, the most numerically abundant life forms on the planet. There are simply more phage particles than all other forms of biology added together. So it's a huge population. It's highly dynamic, with many infections going on an ongoing basis. So much so that it's estimated that the whole phage population essentially replenishes itself in a matter of a few days through the process of infection and reproduction of those particles. It's also thought to be a very old population, possibly going way back to the very early days of life, perhaps 3 billion years. And not surprisingly, then, the phages are highly diverse from a genetic perspective. There are many, many different types of phages genetically, and they vary from each other in all sorts of different ways. Um, that makes some phages easier to find for some bacterial hosts than for others. And we are still very much learning about what this broader population looks like because it is so vast. I just show an example here of a subset of phages that infect bacteria within the phylum of the actinobacteria, just to illustrate their great diversity. If we compare them all and we use phylogenetic tools, we can see that quite often any two phages that you pick up and sequence and compare may have no genes in common whatsoever. The diversity is vast and only a very small proportion of the phage population do we have any actual detailed information about their genetics. It is an area that we desperately need to know more about. When you look at bacteriophage genomes, you can also get some insights into how they have evolved and how they are built. And the overarching rule that you see when you do this kind of analysis is that phage genomes are highly mosaic. And by that, what I mean is that they all share genes and genes get shuffled between them, which means that if you just take pairs of genomes and compare them, you find they have some genes in common and some genes that are not in common. And those genes are being swapped by this very rapid process of evolution amongst the population. So you may isolate, for example, two phages, which are quite closely related to each other. And when you look at their genomes, you see they have some genes in common and some genes that are different. And that genetic constitution is what determines how they grow, which bacteria they infect, and therefore how we can consider using them from a clinical perspective. And so understanding the genome of a particular phage is very helpful in thinking about what its prospects are for a potential therapy. I mentioned that phages have these two basic types of behaviors in their growth. So let's first of all just consider what phages that we would refer to as being lytic as what they do. So in the diagram is showing you here at the top some phage particles. Let's just imagine those are of a phage that is lytic. It identifies and finds a bacterium that it can infect by binding to the surface of the cell. Most of the proteinaceous body of the phage remains on the outside of the cell, and just the genetic information, usually the DNA, gets injected so that it moves from the head of the phage into the inside of the bacterial cell. At that point, the phage essentially uses its genetic code to tell the cell to begin to replicate the DNA so that it makes many more copies of the phage DNA. The phage genes are expressed for making the particles that constitute the body of the phage. And then a process of assembly occurs in which the DNA is packaged inside the head, the tails are attached, and in the final step is what is referred to as lysis, which is the breaking open of the cell and the release of the new phage particles so that they can then go and repeat this cycle of lytic growth. These are the kinds of phages that are useful directly for applications in a therapeutic circumstance. simply because the infection and the reproduction of the phages leads to the death of the bacterium. That process of lysis is really a complete destruction of the bacterial cell to release the progeny phages. Temperate phages have an additional step that they can engage in addition to lytic growth. So the very early stages of infection by a temperate phage are the same. The phage attaches to the surface, injects its DNA, but once the DNA is injected into the cell, there is a decision point, and one of two different pathways can be pursued. One pathway is simply the lytic growth pathway, which is exactly the same as I just described for strictly lytic phages. The other cycle is called lysogeny. And in this lysogenic cycle, the phage DNA typically is integrated to become part of the bacterial chromosome. So the phage can park its DNA inside the bacterial genome where it will reside, and the bacterial will grow happily for many, many, many generations without anything else happening. So all of the lytic cycle genes are switched off. The phage DNA is quiescent, and it's often referred to as a prophage. So the lysogens, the lysogenic cells, are not dead, they're alive. These can represent up to 90% of the infections, and therefore that is not a circumstance that you would want to consider for therapy, simply because the cells have survived the infection. And for therapy, what you want to do is kill them. Now, fortunately, there are things that you can do with this genetically in order to make temperate phages as useful and as available for therapy as lytic phages would be. Because for many phages, the process of lysogeny, the genes required for lysogeny are well known and well understood. And because engineering tools have been developed for altering the genomes and therefore altering the biology of the phages, they've all been developed so that we can simply, relatively simply, go in and inactivate that life cycle, such that temperate phages now essentially become obligatorily lytic phages and therapeutically useful. So the final point I just would like to cover here is the notion, going back to the specificity, that if you are looking for a phage to treat a particular patient infected with a particular type of bacterium with a particular clinical isolate, it's important to have a phage that will both infect and kill that particular isolate. And so we use in our lab the scheme that is shown here for screening strains, and other labs use similar and sometimes some slightly modified schemes. But the idea is simply to grow the patient bacterial isolate, to try infecting it with a panel of phages. We typically do ours using these spot tests, and this enables us to rather quickly and readily identify which phages might be useful for any particular patient isolate. And then we also do challenges where we grow the culture of bacteria, we challenge it with the phage and liquid culture, and ask whether we're able to kill it efficiently. And so that is a process for screening. It's relatively quick and relatively straightforward. In our hands, this process is really an essential process for the compassionate therapy, compassionate use for phage therapies, especially for the mycobacterium strains that we work with. Obviously, in terms of identifying infections for which clinical trials might be appropriate, then this would be cumbersome to do this for every single patient in a clinical trial. So the hope would be that you can assemble a cocktail of phages which would have the ability to infect and kill the vast number of different types of clinical isolates for that particular type of infection. For some bacterial infections, that is now available, but it remains a challenge for other types of bacterial infections. And that is a topic that we would be happy to discuss further. Okay, so I hope that's given you a good but brief introduction to some of the basic biology of bacteriophages and in considerations of how we might use those clinically. And so we'll now go to our post-test question here and see how we did. A few more seconds to put in your answers. Very good. Thank you very much. If there's no more answers, we'll close this out. And now I'll go to an explanation, which is that yes, the correct answer is C. I'm glad to see that most of you were able to answer that correctly. I would just emphasize that answer C was about the necessity for the bacteriophage to efficiently infect and kill the particular clinical isolate. It's true that phages are small, but size is not really an important parameter for the therapeutic utility. The temperate phages are not therapeutically useful as is, but they can be engineered to be useful. As we talked about, bacteriophage genomes carry a modest number of genes, but it's not really the number of genes that's important. It's their ability to infect and kill clinical isolates of the bacteria. So thank you, and we'll go to questions and answers.
Okay, Graham, thanks. Okay, Graham, thanks very much. Great introduction to our biology today to set the stage for how to use them to our advantage. One of the things that I think, one of the points you made is that for hundreds of millions of years, these phages have been uh, basically playing with bacteria and vice versa. A lot of the things that we see in molecular biology weren't described by brilliant PhD scientists. They were just developed by phages as part of their survival game, their squid game with bacteria over time. What are some of these biological tricks that bacteria have learned to resist being lysed by phages?
Yeah, so as you mentioned, Chip, because the bacteria have been assaulted for such a long time by the phages, all sorts of mechanisms for being able to survive that have evolved, and phages have had to likewise develop strategies for continuing to survive and to find new bacteria to replicate in. And so, some time ago, a long time ago now, there was a very important discovery that some bacteria use what we would now refer to as restriction modification systems, where they use restriction enzymes that will specifically digest the phage DNA when it attacks the bacterium, so that it can provide a very high level of survival. And of course, restriction enzymes have gone on to play enormously powerful tools and as for biotechnological um, uh, means. More recently, CRISPR-Cas systems have been described, and CRISPR systems are naturally defense mechanisms that bacteria use to defend themselves. And of course, they've proven to be very effective general tools for engineering strategies. But what's really truly amazing, I think, and has really been an eye-opener for the phage community and the microbial research community over the past few years is the recognition that there are now many, many, many other systems, new systems of all different types of shapes and sizes that bacteria use as what is really part of a very sophisticated immune system that protects them from infection by the phages. I suspect there are many, many more still yet waiting to be discovered. And certainly we know that phages use strategies to counteract these by having anti-restriction, anti-CRISPR, and anti-defense genes that they have evolved. And so all of that complex dynamic is really what has led to the evolution of these very tight specificities of bacteria for their bacterial hosts. So much more to be learned about that process, but it's a critical part of what we need to understand as we consider the therapeutic uses.
So one of the other questions that's come up is as these phages enter and exit bacterial DNA, can they take with them things that we would not want them to take on to patients? Toxin genes, antimicrobial resistance genes. What are some of those considerations about temperate phages?
Yeah, so it's been recognized again for some time, and has been a useful tool sort of genetically just in doing bacteriogenetics, that sometimes phages can package not bacterial DNA, sorry, not just phage DNA or just phage DNA, but can package bacterial DNA. This is a process called transduction. There are essentially two general types of that. One type of transduction is referred to as generalized transduction, where a phage may package really any part of the bacterial genome and then transmit that to other bacteria. And then there's so-called specialized transducing phages, which may pick up just one part of the bacterial genome. It's often been expressed as a concern that you would really prefer to use phages therapeutically that do not do transduction at any substantial level, because if you did, you would at least be exposed to the risk that those phages could take antibiotic resistance genes from a bacterial strain and then transfer it to another bacteria. So you would basically be making the problem worse in terms of resistance to antibiotics. In fact, many phages that can be used therapeutically don't do generalized transduction at any substantial level. Others may well do so. And I think that there's relatively simple strategies that can be used to group phages as to whether they do transduction at any significant level. And if and where there's a choice of phages, you would certainly want to choose those that have limited or undetectable levels of transduction.
So one of the other questions about phage bacterial interactions has to do with biofilms. Why do bacteria make biofilms? What are the functions of biofilms from the bacterial perspective? And people have talked about phages being able to attack these biofilms. Is that something that can be used therapeutically in clinical settings in which biofilms may be making it difficult for traditional antibiotics to clear an infection?
I think there's a general sense now that in nature, as opposed to in our test tubes in the lab, most bacteria probably grow as some types of communities. And those communities of bacteria are often referred to as biofilms, where you have this community of bacteria often attached to a surface. Often it's somewhat differentiated, so different cells within that bacterial biofilm may be expressing different gene expression profiles, and therefore it becomes a fairly complex community. Um, the biofilm usually has not just bacterial cells, but it has components of what is often referred to as the matrix, which is holding the whole community together. That matrix can contain DNA and lipids and sugars, and can be chemically quite complex. It makes it difficult. Many biofilms make it difficult for antibiotics to penetrate into the biofilm and therefore get to the bacteria. And so there are some infections which you find in humans where there probably are biofilm-like communities, and that behavior contributes to the poor responsiveness to antibiotics. At least some phages appear to have evolved to be able to deal quite well with biofilms. Some phages have enzymes actually part of their structures, so-called depolymerases, which will help the phages to eat their way through the biofilm. And therefore, there's considerable promise, I think, that there are some infections for which phages will be the preferred antimicrobial therapies, simply because they'll be able to deal with the cells in those biofilms more effectively than other strategies.
So you've kind of opened the door to discuss making phages better, if that's feasible after the 300 million or so years they've been working on it. Tell us about some of the things people are thinking about in terms of phage engineering. Can you introduce the ability to go after biofilms more efficiently, or to be more lytic, or to have broader host range with molecular tools?
I think there's a number of strategies. First of all, for engineering, there are various tools that you can use in the lab to grow bacteria and phages, such as to engineer their genomes, to change them. Synthetic biology and synthetic genomics offers considerable opportunity, where you can essentially design the genome de novo and then build it, and then have it manipulated to do what you want it to do. I mentioned that there are some phages have anti-defense genes. So you could envisage building a genome either by engineering the lab or maybe building it synthetically, and adding what we would think of as payloads, extra genes that they carry, which would have the effect of broadening their host range, for example. Or maybe you could add ligands, components to the outside of the phage, which would promote the phage to be taken up by particular types of cells, or to be targeted to particular locations or organs even in the body. Um, there's a world of engineering to be exploited there. Um, and It used to be that these types of approaches were sort of technically limited. I think that these days they're limited less by our techniques as much as they are limited by our imaginations as to what we want to build and how we can make phages more useful than the ones that we just identify naturally.
Terrific. So we're really at the verge of a new mode of therapy that the molecular biologists will be able to hopefully tune up, as you have actually with your mycobacterial phages already. The there are a couple of questions here about biofilms that I think we'll get to later, and another one here that asks about what the biological basis for phage specificity might be. You've addressed some of this, but why don't you talk about what determines whether a phage can go after a particular bacterium?
Yeah, no, it's a good question, and I think, as I tried to intimate it earlier, that there's probably a large multiplicity of effects, but it's also helpful to think about splitting those up into different stages of the phage infection. If you remember, in those cycles, the first thing that has to happen is that the phage, sorry, has to be able to recognize a particular bacterium in order to be able to infect it. And so usually we think of that as the phage recognizing and binding to a particular type of receptor molecule that's on the outside of the cell. And therefore, the presence or absence of a particular type of receptor can be and often is a major, if not the major determinant as to whether a phage can infect that particular type of cell. If there's no receptor, the door is closed, there's no way of getting in. That's really the first level of defense, if you like, that bacteria can have. The second are all of these other defense types of systems that really are effective only after the DNA has been injected into the cell. So those systems that we talked about, like restriction enzymes, for example, CRISPR-Cas, and actually many of the other of the natural defense system are all ones that act after the DNA has entered the cell. And we know now that phages often use receptors on the surface of the cell that are important for the biology of the cell. So I think that in general, phages over their 3 billion years of evolution have had a tendency to be dependent upon the host, the bacterial host functions that it's really hard to get rid of, and therefore minimizes the opportunities for resistance in the natural environment, even though we know that resistance can occur quite readily in laboratory settings.
All right, thanks very much. I think we've addressed all the live questions, and we can move on and spend a bit of time talking about some of the potential clinical applications of phages, and then come back and have some more joint questions and answers. Thanks very much, Graham. That was terrific.
Thanks, Chip.
All right, am I on the presenter view, or am I on the slideshow view, folks?
On the slideshow right now,
Okay.
Chip, if you would go to
Let's
the
yeah,
presenter view
we'll get to that then. That's great.
down at the bottom. There you go.
Okay. So I'm going to spend a bit of time talking about some of the clinical applications of bacteriophages, and we'll briefly discuss some of the recently completed clinical trials that I think are beginning to shift us in the direction of understanding how best to move forward and to think about these for what they really are. They're just living antibiotics. And a lot of the things we've learned about how to develop antibiotics can be applied productively to the therapeutic development of bacteriophages. My disclosures are shown here, and my learning objectives are fairly straightforward. I'll talk about some of the most promising clinical applications, a little bit about some of the issues related to bacteriophage pharmacology, and then, as I mentioned, I'll talk about some of the recently completed clinical trials. So here's your pretest question. Let me let you look at the question and the answer box and have a couple of minutes to decide what direction to go with this one. Okay, so the most uh, prevalent answer is C. Let's go on and move to the next section of this, and we'll come back to that later. Thank you. So this is one of the graphics from the Global Burden of Disease group that has really led the way in helping us understand the implications of antimicrobial resistance, both today and in the future. I encourage you to have a look at it. The Lancet has published much of this, but here you can see the global landscape projected for 2050 in terms of where the problems will be. Things that look red and orange are worse than things that look blue. And what you can also see on the right panel of the slide is that as we move forward, the number of deaths globally are clearly going to continue. And much of this will be focused in the older patient populations. Now, one of the problems we've had is that although we had for a long time just a row of fantastic advances in developing traditional antimicrobials for about a 40-year period, we had new antibiotic classes coming out every couple of years. And these antibiotic classes had a major impact on our ability to treat people with bacterial infections. And I think by the late 70s, we assumed this would continue. But you can see that since that time, the pipeline has really gotten quite dry. And a lot of what we've seen have been tweaks, a methyl group here, a methyl group there, adding a few, a little slightly broader spectrum against Proteus morgani, but not a lot of fundamentally new antibiotics. In the meantime, below the line here, you can see in the same timeline, the date at which bacteria from the clinic began to show resistance to the antibiotics being used. And while we continued to try to develop new antibiotics, bacteria have continued to outpace us in terms of developing resistance in face of the antimicrobial exposures they've had in patients, in veterinary medicine, and in agriculture. Now, the other thing we've also begun to think about, you can imagine trying to come up with bacteriophages that help us get out in front of this. A question that was asked earlier has to do with other aspects of phages that might be beneficial in terms of treating difficult to treat infections. And it has to do with, as Dr. Hatfield discussed, the ability of phages to attack biofilms and the ability of actually molecular biologists to enhance this capability. This has led to the second major application of phages up to date, in addition to antimicrobial resistance, and that is to turn phages loose, usually with antibiotics, on infections that involve biofilms. We think about biofilms primarily being present on the surface of implanted devices, but there are also biofilms present in other anatomic locations, for example, osteomyelitis on the surface of heart valves that become infected, and basically in any bacterial population in which infection slows and they begin to grow as colonies. Now, phage therapeutics have been around for a long time, and I think it's important to put this into perspective to get an idea about kind of where we are. Phages were first described in the 1910s by a couple of groups almost simultaneously who noted that if you took bacteria and put them onto an agar plate as a lawn and then sourced in some cases river water and filtered it through a filter that would allow things smaller than bacteria, in those days known as viruses, through the filter, and then put that filter on the lawn, you could see holes develop. Something was eating the bacteria, hence the name bacteriophages was coined. It took a while to figure out these were actually viruses, and there was a period of giddy optimism for about 15 years in the West, where bacteriophages were thought to be the answer of bacterial infections. This was before we had widespread use of antibiotics, before the sulfonamides came along. And phages were actually sold as cocktails, albeit poorly characterized, for specific infections. For bacticolophage, for example, for diarrhea, rhinophage for upper respiratory infections. And these consisted of kind of empiric collections of phages grown on groups of bacteria that sometimes were viable when the phages arrived in the pharmacy, but often were not. By 1935, it became clear that phage therapy was hit and miss in terms of the way we were using them, and antibiotics began to be developed. And what happened in the West was we moved in the direction of antibiotics. In the East, particularly Georgia, the former Soviet republics, bacteriophage therapy continued, in part because antibiotics were expensive. And to this day, much of what we've learned about bacteriophage therapeutics has come from those locations. There was a renaissance in phage therapy in the mid-2010s when a couple of things happened. First, we recognized that we were not keeping up with bacterial evolution with antibiotic development. We began to look for other tools. And secondly, new biotechnological approaches were developed that allowed us both to identify phages efficiently and to purify them so that they could be given parenterally as we do antibiotics. For the last 10 years, we've been using them primarily in single patient experiences under regulatory oversight, so-called single patient INDs. But what we're seeing increasingly is organized clinical trials that are opening the door to phage therapeutics, moving to mainline medicine, which I think is where we all hope it will go. Case reports have been exploding. This is just a English literature case reports in PubMed, sorry, over the last 10 years. And you can see that things have picked up starting about the mid 2010s. And in Europe, phage therapy is thriving. This is a series published by Jean-Paul Pernet of 100 consecutive cases at their center in Belgium. And I would encourage you to have a look at this Nature microbiology paper because it's quite comprehensive in terms of explaining what types of infections they're treating and providing some insights into some of the questions that we still need to address more directly about the induction of antibodies to phages when they're given parenterally, the frequency with which bacteria that are under treatment develop resistance to phages, and a better sense of how often phage therapy works in these sorts of settings. In the US, it's become increasingly recognized that phage therapy may have clinical benefits and may be beneficial to some patients. In our center called IPATH at UC San Diego, we have a web-based tool that allows people who are interested in phage therapy for infections that may not be responding to contact us. And to make a long story short, we've received about 3,000 requests over the last 7 or 8 years for assistance with patients who may need phage therapy. Many of them don't. Turns out they may have infections that are caused by susceptible bacteria or even viruses. But we've had about 15% of these turn out to be patients we think are worthy of trying to find a phage that would be active. We're able to do this about half the time, and only about a third of the time do we actually get to patients who get treated. What are the organisms that we run into that people call us about? Very much the same ones you'll see in the Pernet series from Europe, Pseudomonas, Staph aureus. Now, Staph aureus requests are usually around implanted prosthetic devices, the biofilm indication that I mentioned. And then enteric organisms that carry ESBL genes. Acinetobacter is a common organism as well, and then a smattering of others. And further down the list, you will see non-tuberculous mycobacteria that is almost exclusively handled in the US by Graham and his group. Now, how do we get patients involved and get therapy to them? Well, it all begins at the bedside when a physician recognizes a patient has a difficult to treat infection. The clinical microbiology laboratory isolates the organism, the antibiogram is generated, and it becomes clear that antibiotics may not be likely to be successful. The clinical microlab then speciates the organism, and at this point, someone who has some insight into phage therapeutics is involved. In our case, we usually interact with the physician who is seeing the patient and discuss whether or not in this particular clinical indication phage therapy might be helpful. By this time, we know whether the organism is, for example, Staph aureus or an E. coli. There are a number of academic laboratories around the country, each of which has essentially a phage specialty. For example, at Baylor, there's a lot of work with enterics. Here, at work with Enterococci. Graham's group works on non-tuberculous mycobacteria, and we try to do essentially a matchmaking to get that organism to the right laboratory where screening is performed, and if a phage is found, clinical grade phage is produced. The clinician then has to get in touch with the FDA and describe to the FDA both the clinical scenario and how the phage was made, obviously with the support from the laboratory that does it. If the clinical situation is emergent, the clinician then describes the case to the FDA as being something that needs to start emergently. The FDA can give approval essentially over the phone with no engagement of the local IRB. The clinician then, with the consent of the patient, can start therapy and notify the IRB later within 5 days locally. If it's a more elective situation, the local the physician puts together an IND, actually a relatively simple process. The local IRB then opines, and the patient then can be offered therapy. This process takes some time. The major two bottlenecks are finding phage in the academic laboratories. It's much easier for some organisms than for others, because many of these times these days, laboratories have a pretty comprehensive collection of phages for specific organisms, and it's just a matter of doing the screening. Other times they have to go out in the environment, which can take a long time, weeks to months. And then the second bottleneck is preparing clinical grade phage, and we can talk a little bit later about what that requires, but it essentially means purifying the phages with some bacteria getting rid of endotoxin, and making the phages appropriate to be given to humans as therapeutic agents. Now, how often does this work? On the right side of this, in this 100-patient experience from Belgium, Dr. Pernay, the bottom line is about 3/4 of those patients seem to have clinical benefit. In our experience, again, it's about 55%. Now, these results should be looked at skeptically, because these are basically follow-up questions to clinicians administering them with their impression about how patients with a broad variety of different types of infections responded, often without rigorous microbiological endpoints. But what I'm getting at here is that, at least from the perspective of the treating physician, half to 3/4 of the time, this approach leads to a beneficial clinical effect. In the case of non-tuberculous mycobacteria, this is a very nice series that Graham's group put together in clinical infectious diseases a couple of years ago. You can see again the clinical outcomes, partial, complete treatment success in about half the cases. So the bottom line is, when approached this way, there has been enough evidence of clinical utility that it has encouraged people to try to proceed in a more organized way into clinical trials. So why can't we keep doing this? Well, the major problem is that we'll never get to the scale needed to be able to deliver phage therapy to all the people who need it. I was talking earlier about millions of people who have antimicrobial and biofilm-associated infections. In the last 8 years, I've been talking here about 150 or 200 cases that, with a lot of people working, a lot of expense, have managed to be treated. We can never scale this up out of academic laboratories. It's going to need to be done the way we do other therapeutics. We're going to have to find ways to commercialize this and to find ways that it can be supported sustainably. We also need to have more knowledge about how to use phages in the clinic. That'll require clinical trials. And we're learning a lot about technical innovations that will make phages more useful in terms of engineering, how to use them together with antibiotics, and how best to predict their clinical utility with phage susceptibility testing in vitro. Now, clinical trials of phages have picked up as well. These are ones that are listed on clinicaltrials.gov, the date they're listed. And you can see this began to pick up again about 2020, with between 5 and 15 new studies announced each year. The 2025 data are obviously just until earlier this spring. Now, what have we learned from some of these studies? This is a study that was done in people with cystic fibrosis who were chronically shedding Pseudomonas, and they were administered an aerosolized cocktail of anti-Pseudomonas phages. And one of the clinical endpoints was the colony count of Pseudomonas in sputum 15 days after a 5-day course of inhaled phage. And what you can see is those who received the active phage cocktail compared to placebo had on average about a 2 log 10 colony count decline in Pseudomonas over this period of time. So a clear benefit from the microbiological perspective in this clinical trial, from which we learned both one way to assess microbiological endpoints and the fact that when phages are given to people with pulmonary infections by an aerosolized route, they indeed get to the site of infection and can have a beneficial microbiological effect. This study was not powered for clinical benefit, but having learned about this and having done some dose ranging to get to this, the group is now poised to be able to move on to more clinical endpoint trials. This came from a study that was published in Lancet Infectious Diseases last year of people with recurrent urinary tract infections. What we learned from this study was a couple of things. First of all, that when you give phages by an intravenous bolus, you get a peak of level of phages in the plasma about an hour later. And then by 4 hours later, this, if you look at the x-axis here, this 1.0, 1.1, 1.2, these are hours after infusion on day 1, and then 2.0, 2.1, 2.2 hours after infusion on day 2. These were given on 3 days. So you can see that by 4 hours, most of the phage given by bolus have declined and have gotten below the limit of detection. You can see, however, if you increase the dose of phage and give it as an infusion, persistence in the plasma is longer in the right panel. Now, in this study, you can also see that when the phages also appear in the urine when given intravenously and into the bladder, but they decay over time as well. These types of studies can begin to provide insight into how to design clinical trials with clinical benefit. And most interestingly in this trial, which was done by randomizing people with recurrent E. coli UTIs to trimethoprim sulfa versus trimethoprim sulfa plus this anti-E. coli phage cocktail, the microbiological success rate was over 80% in people who had trimethoprim resistant E. coli. So my point here is this trial clearly demonstrated microbiologic activity in the absence of effective antibiotics in urine in this trial that was designed in a way that allowed these clinical endpoints to be determined. This trial also has set the stage for phase 2-3 trials to try to understand best how to use them in clinical medicine. A study that has not yet been presented beyond press release phase is one that was conducted in patients with diabetic foot osteomyelitis. These were people who had Staph aureus cultured from bone debridement, not from swabs, but from debridement of bone with diabetic foot ulcers. They received optimized standard of care antibiotics or a single dose of intravenous phage with topical phage for 3 months. And again, both microbiological and clinical endpoints were accelerated in the active treatment arm in this study of diabetic foot ulcers. And the final study that I'll mention is one that again was just top-line results were just announced about 6 weeks ago. This was a study of people with complicated staph sepsis who were randomized to receive or optimized antibiotics plus a anti-staph phage cocktail. And to make a long story short, when one looked at again investigator-assessed responder rates And also with the bacterial clearance, the phage therapy was more successful than antibiotics alone. This study obviously is going to stimulate more studies with phage therapeutics. So we now move to a day in which we have data from clinical trials that show microbiological or clinical benefits in several different indications that are being studied for phage therapeutics, used in some of the anecdotal use over the last 10 years. And that I think we'll now see more and more organized clinical trials moving forward. I didn't discuss today a study that's still ongoing, trying to use phages orally to affect the microbiome in people who are colonized with fluoroquinolone resistant E. coli who are awaiting stem cell transplantation, trying to debulk the GI tract and reduce the likelihood of sepsis. But phage, in addition to being used for specific therapeutic indications for individual infections, are also beginning to be thought about as potential ways to affect the microbiome. So to conclude, where are we now? Not too long ago, we had a lot of discussion about phages and how they might be used. And what we've seen over the course of the last 18 months is that we've begun to see more water in the lake here, and that all of these boats are beginning to rise as we're learning more from one study that helps us prepare additional studies with multiple different clinical indications. So I think we're really now at an inflection point where phage therapeutics will increasingly be seen as a way forward in antimicrobial resistant infections. So let's move back to the post-test question, the one you saw before, and see if you can improve on your outcome, which was pretty stellar to begin with. So let's let people address this, and then we'll have a few minutes for questions and answers. Okay, pretty good. 98% of you answered the C, which is reflects the data that I mentioned about the uh, uh, staph sepsis study that was just recently unblinded that we just heard about. And the other answers, you can see the reasons that they weren't optimal because of the uh, results shown here. So let us, rather than obsessing about the answers that weren't correct, since all of you, virtually all of you got the right answer, move on and let Graham moderate a brief question and answer session before we finish. Graham?
Thanks very much, Chip. Super. Let's just, if we could just start with a question, Chip, about some consideration of the difference in the regulatory considerations for the use of phage versus antibiotics.
Yeah, it's interesting. The regulators have been cited for a long time as being the impediment here, but I actually think, as was recently said, the problem isn't in the stars, the problem is in us. And that is what regulators are looking for, is they're looking for any kind of a therapeutic that could be shown to benefit clinically a patient with a disease um, that needs an intervention. So what needs to be demonstrated to the regulators is that you have a phage or a phage combination that will provide clinical benefit to a patient with an infection or with a microbiome that needs manipulated. You have to have a clinical indication and move forward. Now, one of the issues people have raised is, well, they want these phages to be so pure. Well, obviously, that's what you want when you're having a pharmaceutical given to somebody. You don't want to have somebody say, I've got this antibiotic that's almost pure. Let's just go ahead and use it because it's so much trouble to purify it. And then you're worrying when you have side effects, whether it's the product or what goes with it. So it's very important these phages be made to the high standards that that we would use for any pharmaceutical. We've been doing this forever with monoclonal antibodies and other things made in bioreactors. No reason not to be done with phages. Then the final issue is how do you characterize them? As you pointed out, these are dynamic little beasts that are always chasing bacteria, and they will evolve over time in patients. But what we can characterize is uh, which individual phages are in a cocktail, what their spectrum of activity is, and with susceptibility testing, what we predict their activity to be clinically. Now, this latter aspect will take some time to firm up. It's taken us 40 years to make antibiotic susceptibility testing more predictable than it used to be, but we're moving that direction here with phages. So I think what we're going to see is the same regulatory framework that we see with antibiotics. We have to first define the pharmacology. We have to define toxicity. This is something that the FDA is not worried about with phages because we frankly don't see toxicity with phages if they're purified and given topically, intravenously by an aerosol route. We then have to demonstrate activity, and then I think we're going to see clinical indications approved by regulatory bodies. So I think this is a paper tiger that just requires us to think about how antibiotics have been developed and use the same principles.
Great. A question from the audience. If I'm a clinician and a patient comes to me with a question, they've read all about phage therapy on the internet or on social media, what steps can they take to either try to look for possible phage solutions, or perhaps more ambitiously, if they wanted to establish a, a structure or an organization at their own institutions where they could do these things more routinely, what would be a good starting point?
Well, I think a good starting point would be to read John Paul Pernay's paper that was in Nature Microbiology. Graham and I have a couple of things that we put together as well that provide some background into phage therapy that give some insights into clinical applications that might be useful. When somebody comes to you with a patient, the first thing to answer is to address is whether or not phage therapy is the way to go. And this usually is a gating question. Could you solve the problem with antibiotics? That's worth asking because it's certainly a lot easier to write an order for an antibiotic than to run around and look for a phage and deal with regulatory issues. Once you've determined that either because of the antimicrobial spectrum or because of failure because of a biofilm that a phage would be useful, you then have to find someone who can make it for you. We're happy at iPath. If you want to email us, we can help you to navigate you to the right place and find people who will make it for you and give you advice about how to administer it over time. I think you'll develop expertise to do this yourself. You have to realize a lot of the advice we've been giving has been empiric. We generally use 10 to the ninth plaque forming units because that's what we've used. The Europeans have been using 10 to the seventh plaque forming units as their base phase dose, and both seem to work. So we've got a lot of things that need to be worked out in clinical trials. But as you begin to have experience with patients and follow the literature, I think you'll feel more comfortable doing it. Now, if you have a microbiologist in your institution who wants to get into this with you, that's even better because they can work with you to set up phage susceptibility testing. A clinical microlab can do that. Basic scientists like Graham might find an interest in developing a capability of developing a phage library for specific organisms and ability to produce them for clinical use. And pretty soon you're off and running with having a center of your own. I would encourage you, though, to also keep your eye open for clinical trials because getting patients into clinical trials is how we learn how to use phages. And as in the case of HIV therapeutics, we learned a lot as clinicians about how to use these drugs by participating ourselves in clinical trials. And I think phage therapeutics will have the same learning curve for those of you who want to get into using them in clinical trials.
Thanks, Chip. We have a question about host immune responses to bacteriophages when used therapeutically? To what extent could they be beneficial? To what extent could they be detrimental to the phage therapy?
That's a great question. It's one that we're learning about as we go. Um, in that series of John Paul Pernays, they had follow-up plasma on less than 20 patients to be able to look for phage antibodies. And they found them in parenterally administered courses of therapy about half the time. And in about half the time, they thought that the antibodies might compromise phage uh, therapeutic responses. Graham's situation, in which non-tuberculous mycobacteria have been used therapeutically. Let you um, um, unleash your own lightning and thunder, Graham, and tell them what you found in that scenario.
Well, I would just say that we've seen what we've interpreted as both um, detrimental aspects and otherwise, in the sense that there are certainly patients, one in particular that we've reported, where we observed quite a strong antibody response that was neutralizing to the phages in vitro, and that appeared to correlate with the timing of a less robust effectiveness of the phages therapeutically. On the other hand, there's a number of other cases where we've seen, I think you would describe them as favorable clinical outcomes after treatment with phages for many months, even though the patients mounted a robust neutralizing antibody response. So we've seen both things, and I think just emphasizes the need for controlled clinical trials where you can understand these variables in a much more meaningful way.
Yeah, it's not a reason not to use phages because we don't see any evidence of toxicity to the patient when these do develop. It's something that I think we need to understand in terms of their impact clinically. You can envision a situation where phages get to a place where bacteria or mycobacteria are living, replicate in that location, and jump from organism to organism with antibodies present in the plasma. They're irrelevant to what's going on at the site of infection. So I think there are a lot of dots we still need to connect to really be able to answer that question intelligently.
Then we have a question about what happens to the phages when you administer them therapeutically? Do they continue to circulate? Are there documented half-lives? What are the pharmacokinetics like relative to, say, typical antibiotics?
Phages are picked up by the reticuloendothelial system. If you label them, you'll find what's left over after they've been picked out in the spleen and in the liver and the reticuloendothelial system. They get picked out pretty quickly over the course of the first couple of hours after an infusion when you give them intravenously. What then happens is they get recycled, the torn apart recycled, and their amino acids and nucleic acids are used by the cells that pull them apart for other purposes. So they're basically just used as one of the many things we scavenge from our environment for our own use. Now, in the environment, they live for a longer period of time. You can isolate them, for example, from sewage water if you go to a sewage treatment plant. Um, they're quite stable. Most of them are quite stable in physiological solutions at 4 degrees centigrade. So you can, in those situations, stabilize them with the right buffers and use them like you would a biological product for therapeutic use. But in the body, our RE system is looking out for foreign materials, viruses, and this is one of the things they pull out. So they don't stick around forever.
We just have two more quick questions here, Chip. The first is, and it may be pertinent to a point that you were just saying, which is there some utility to going to places, including hospitals, where there are known high levels of AMR and looking specifically and targeting those areas for the isolation of new phages, which may be particularly useful clinically?
That's a great question. The answer is absolutely yes. Phages are better at finding their bacteria than we are, and we should take advantage of their ability to sniff out bacteria they can live on. And so a very good place to find phages may even be in the GI tract of people who have these multi-drug resistant bacterial infections. If you're thinking about staph phages, look on dressings from people who have staph wound infections. Phages go to where their food is, and that's really an important thing to remember in your phage hunts. Bacteria get around the globe pretty well, and when you look at some of the multi-drug resistant bacterial infections that you see, for example, in India, the next thing you know, they're in the US and vice versa. So we have a fairly global range of bacteria and phages that go with them. And it's not always necessary to go to Thailand, for example, to find phages that will be active against a multi-drug resistant organism from Thailand. Those phages may well be present here as well. The most important thing is to have the bacterial organism itself on which you can do screening.
Great. So just thanks, Jim. Just the last question is it the question was whether it's always necessary to screen a patient isolate for phage sensitivity, or are there phages that infect all the clinical isolates? And then the other half of that was just about whether there are clinical trials on the horizon for some more problematic pulmonary infections like the NTM infections.
Most of the time we do phage susceptibility testing because it helps us, at least in theory, have phages in the regimen that you're giving someone that are active against their organism. Now, one big caveat is that how bacteria grow in resource-limited settings, and by that I mean sitting in a big colony of bacteria struggling to find nutrients in a heart valve, for example, is very different from the way we set them up to grow in log phase in the laboratory, where we do our phage susceptibility testing. And so just like with antibiotic resistance testing, we need to do clinical validation to see whether or not the techniques we're using in the laboratory predict clinical outcomes. There has been a major effort for the last 50 years to do that with antibiotics, to come up with what the breakpoints are. The Europeans are beginning to do this for phages. We haven't really started that in the US, but I think it's a critical part of clinical trials to understand the extent to which phage susceptibility testing predicts or doesn't clinical outcomes. That's a great question.
Thanks very much, Chip. Well, that concludes the Q&A for this section, and so we'll go to you for the final closing comments, Chip.
Okay, so I wanted to thank everybody for both your attendance and the great questions that you sent to us. If you have other questions, feel free to send them in this direction. We'll try to get back to you about them. Please also provide your feedback about how to do this better in the future. We are always looking for ways to improve both the content and the mode of delivery. So please feel free to tell us how we could have done better. The ways to get your CME are shown for you there. And we also want to give you a special invitation to a conference on bacteriophages that Graham and I will be hosting with support from IAS-USA in Washington, D.C., October 12th to the 14th. This is a conference that we've modeled on those of you who know HIV, CROI. We want to have basic scientists, translational researchers, clinical researchers in the same place to present a wide variety of multidisciplinary phage-related science to accelerate the field in a forum in which people can interact freely and see the most recent phage research presented. We've had the first of two abstract deadlines with nearly 100 abstracts having been adjudicated. We have another abstract deadline coming up. You don't have to submit an abstract, obviously, to come, but we do hope that many of you will come, because I think a lot of the questions that you've had in the question and answer period will be ones that will either be addressed at this meeting or ones that people will be wanting to know more about. So please please come to this meeting. We have both options for in-person attendance, which we hope people will take advantage of because these interpersonal interactions are really productive. If you can't come, we're working on putting together some virtual options. There will also be there is a podcast that Dr. Hatfield has done, and you can see this on our Spotify, Apple Music platforms. This was led by Mike Sag. And another one upcoming is one that is of broader interest, having to do with how vaccines get approved in the U.S. and the story of respiratory syncytial virus that Yvonne Maldonado, one of the real members of the real CDC Committee on Vaccines, will be participating in with Mike Sag. We think it's quite topical in view of what's going on with vaccine development and availability these days. So thanks again for participating. We hope to see you in future IAS-USA activities, and we also hope that many or all of you will come to be with us in Washington in October. Thanks very much.